students. It's another time that we are meeting in our normal Elimika program. Our class is still the same, Form 5, and we still have our topic of nutrition. My name is Fortunato Schiobia. Welcome. Well, my student, you can remember last time we met, our discussion ended up with describing the two categories of plants that are classified according to their immediate product of carbon dioxide fixation. That was C3 and C4 plants. Now, can you remember what we did say? Briefly, what we did say about the two categories of the plants? What did we say about the C3 plants, and how did we define the C4 plants? Hopefully you remember, we did say that the C3 plants are those plants whose immediate products of carbon dioxide fixation is a 3-carbon compound by the name of phosphoglyceric acid, or simply glycerate phosphate. Whereas the C4 plants, these are those plants whose immediate product of carbon dioxide fixation, the primary product of carbon dioxide fixation, is a 4-carbon compound that is oxaloacetate or oxaloacetic acid. Now, having seen the meaning of the two categories of the plants, let us see the differences that exist between the two categories of the plants. I mean, differences between C3 and the C4 plants. Now, what are the structural, physiological, as well as behavioral differences of the two types of plants? One, let us have the C3 plants on the side and the C4 plants on the other side. Well, from our definition for the two types of plants, we have said that these C3 plants are those plants whose immediate product of carbon dioxide fixation is a 3-carbon compound, that is PGA, and C4 plants are those plants whose immediate product of carbon dioxide fixation is a 4-carbon compound, that is oxaloacetic acid. So one, the immediate product of carbon dioxide fixation is phosphoglyceric acid, I mean a 3-carbon compound, whereas here the immediate product of carbon dioxide fixation is oxaloacetate. This is a 4-carbon compound. But secondly, in the C3 plants, there is only one type of chloroplast, that is a bundle sheath chloroplasts. While in here, in the C4 plants, there are two types of chloroplasts. I mean mesophyll and bundle sheath chloroplasts. And because of the presence of only one type of chloroplast in the C3 plants, carbon dioxide fixation occurs once. So carbon dioxide is fixed once. While in the C4 plants, the fixation of carbon dioxide occurs twice. Carbon dioxide fixation occurs twice. Firstly, in the mesophyll, but secondly, in the bundle sheath. But also, the substrate for carbon dioxide fixation in the C3 plants is a 5-carbon compound that is ribulose biphosphate. While in the C4 plants, the substrates for carbon dioxide fixation are 1, phosphoenolpyruvic acid. This is a 3-carbon compound. This fixes carbon dioxide in the mesophyll. But also, there is refixation of carbon dioxide, as we're going to see, by the normal ribulose biphosphate. This occurs in the bundle sheath. But also, apart from having ribulose biphosphate fixing carbon dioxide in both and only PEP found in mesophyll, the enzyme for fixation of this carbon dioxide in the C3 plants is ribulose biphosphate carboxylase, or it is, in short form, you can call it Rubisco. But in this case, the enzyme which catalyzes the fixation of carbon dioxide are of 2 categories again. For the phosphoenolpyruvic acid carbon dioxide fixation, we have phosphoenolpyruvic acid carboxylase. But for ribulose biphosphate carbon dioxide fixation, we use the enzyme used is the same ribulose biphosphate carboxylase. This enzyme ribulose biphosphate carboxylase, if compared to phosphoenolpyruvic acid carboxylase, as far as the power to combine or to fix carbon dioxide is concerned, the phosphoenolpyruvic acid is more powerful in fixing carbon dioxide than ribulose biphosphate. I mean, it has a higher affinity for carbon dioxide than ribulose biphosphate. And for that reason, this is more efficient. And for that reason, therefore, the C4 plants are tolerant to low carbon dioxide concentration. They can survive well in areas with low carbon dioxide concentration compared to the C3 plants. So these are not tolerant to low carbon dioxide concentration, while the C4 plants, they are tolerant to low carbon dioxide concentration. And because of that, the C4 plants, in the C4 plants, photosynthesis is less efficient, and therefore the yield is low. Photosynthesis is less efficient, and therefore the yield is low. In the C4 plants, photosynthesis is much more efficient, and therefore the photosynthetic products are high, more efficient, and therefore the products is high. We have seen that the C3 plants that in the C3 plants, the enzyme used is ribulose biphosphate carboxylase, but its affinity for carbon dioxide is lower compared to phosphoenolpyruvic acid carboxylase. And therefore, in areas where the concentration of carbon dioxide is low, but the concentration of oxygen is high, the enzyme ribulose biphosphate carboxylase shifts. Instead of fixing carbon dioxide, it catalyzes the combination of the substrate ribulose biphosphate with the oxygen. And this process is referred to as photorespiration. So in the C4 plants, C3 plants, photorespiration occurs, while in the C4 plants, no photorespiration. Well, as I have said, that in the C3 plants, photorespiration occurs. I mean a process by which ribulose biphosphate, instead of catalyzing the fixation of carbon dioxide to ribulose biphosphate, it catalyzes the combination of ribulose biphosphate with the oxygen because its affinity for carbon dioxide is low. But for the C4 plants, no matter how low concentration, no matter how low the concentration of carbon dioxide is, still phosphoenolpyruvic acid carboxylase will catalyze its combination with phosphoenolpyruvic acid. Now, those are the differences between the C3 and the C4 plants. Suppose you are given a question that show or give the differences between Hatch-Slack and Calvin pathways of carbon dioxide fixation. Well, the mechanism by which carbon dioxide is fixed in the C4 plants is referred to as a Hatch-Slack pathway, as we are going to see soon. But as we saw for the C3 plants, the mechanism that is used to combine, to fix carbon dioxide, according to Melvin Calvin, this is a Calvin pathway. Now, the differences between the two categories of the plants, some of them are answers of this question. In the C3, in the Calvin pathway, for example, the substrate for carbon dioxide fixation is ribulose biphosphate carboxylase. Whereas in the C4 plants, in the Hatch-Slack pathway, phosphoenolpyruvic acid is a substrate. The enzyme in the Calvin pathway is ribulose biphosphate carboxylase, whereas in the Hatch-Slack pathway, the enzyme is phosphoenolpyruvic acid carboxylase. Some of these cannot, some of the differences between, these listed differences are directly going to the plants themselves. They cannot be applied to the pathways. For example, we cannot talk of the Hatch-Slack, the Calvin pathway being not tolerant to low carbon dioxide concentration. This is going directly to the plant. So choose some of the differences from these between the two categories of the plants, those which meet the requirement of the question for the two pathways. Now, we have seen the differences between the C3 and the C4 plants. We switch over to the mechanism of carbon dioxide in the C4 plants. We did see the mechanism of carbon dioxide fixation in the C3 plants. When we were discussing about the dark reactions of photosynthesis. Now we have the mechanism of carbon dioxide fixation in the C4 plants. Hello, student. Having seen the differences between C3 and the C4 plants, now let us see the mechanism. Let us look at the mechanism by which the C4 plants fix carbon dioxide. Well, according to two scientists, Hatch and Slack, in 1966. The fixation of carbon dioxide in the sea for plants occurs twice. We have seen in the differences between C4 and C3 plants that the carbon dioxide in the C4 plants occurs twice. Can you remember the parts in where carbon dioxide is fixed? Definitely you can. First, carbon dioxide is fixed in the mesophyll, but secondly, it is fixed in the bundle sheath cells, the mesophyll cells and bundle sheath. These are cells in the leaves. And this fixation involves 4 steps. The first one is fixation of carbon dioxide in the mesophyll. The second one is shunting of malic acid, that is the malate shunt. The third step is regeneration of carbon dioxide acceptor. That is the phosphoenopyruvic acid. And lastly is refixation of carbon dioxide in the bundle sheath. Refixation, carbon dioxide. We have fixation of carbon dioxide and refixation of carbon dioxide. And this is clarifying the fact that carbon dioxide in the C4 plants is fixed twice. Now let us see the first step, that is fixation of carbon dioxide in the mesophyll. Well, in the mesophyll cells, the substrate for carbon dioxide fixation is phosphoenopyruvic acid. And this substrate fixes carbon dioxide in the presence of water, the enzyme being phosphoenopyruvic acid carboxylase, and the product formed is oxaloacetate. This is a 4-carbon compound. Now, the formed oxaloacetic acid, or simply oxaloacetate, will undergo amine reduction. The formed oxaloacetate is reduced by hydrogen from nicotine amide adenine dinucleotide phosphate. That is, here we have in its reduced form. Then it will reduce oxaloacetic acid into malic acid. That is nicotine amide adenine dinucleotide phosphate. So we have a reduction by addition of hydrogen of oxaloacetate so as to form malate. Again, this is a 4-carbon compound. So this is how carbon dioxide is fixed in the mesophyll by the enzyme phosphoenopyruvic acid carboxylase. Remember, we have said that this enzyme is more efficient compared to ribulose biphosphate carboxylase, and therefore it is advantageous over the Rubisco because of two reasons. One, it has a very high affinity for carbon dioxide, and therefore even if the concentration of carbon dioxide is very, very minimal, this enzyme is capable of fixing it, unlike ribulose biphosphate carboxylase, whose fixation requires a very large or relatively large amount of or concentration of carbon dioxide. But secondly, the phosphoenopyruvic acid carboxylase is not competitively inhibited by oxygen. And that's why in the C4 plants, photorespiration does not occur. Remember what we have said when we were discussing the differences between the two categories of the plants. I mean the C3 and the C4 plants. From carbon dioxide fixation in the mesophyll cells, we have the next step, that is malate shunt. The deformed malic acid in the mesophyll cells is now shunted, is now transported, is now passed into the cytoplasm, or into the chloroplast of the bundle sheath cells. This malic acid passes through the plasmodesmata, thread-like structures that join neighboring cells of the plants in order to ensure a living connection between them. So, from the cytoplasm of the mesophyll cells, malic acid is shunted into the chloroplasts of the bundle sheaths. In these bundle sheath cells, malic acid undergoes two reactions. One, it is undergoing oxidation by the hydrogenation. But secondly, it is undergoing decarboxylation, where it loses carbon in form of carbon dioxide. In other words, this malic acid is oxidatively decarboxylated to form pyruvic acid. So we have malic acid, a 4-carbon compound. This is oxidized. We have nicotine amide adenine dinucleotide. Then we have its reduced form. Then carbon dioxide is lost. Then the resulting or simple pyruvate, a 3-carbon compound. So this pyruvate is formed in the bundle sheath. And then after formation of pyruvate, there is regeneration. Regeneration of a carbon dioxide acceptor. In this case, a phosphoenopyruvic acid compound. The formed pyruvic acid in the bundle sheath is used to regenerate phosphoenopyruvic acid. We have pyruvic acid or pyruvate used to regenerate phosphoenopyruvic acid. There is an addition of an inorganic phosphate. And this inorganic phosphate comes from ATP. So we have pyruvate. This is phosphorylated by an inorganic phosphate from adenosine triphosphate. Take one inorganic phosphate. Then here we have ADP. So the resulting product is phosphoenopyruvate. So we have formed again the carbon dioxide acceptor. It is important that at the end of the cycle, we form the carbon dioxide acceptor so that the acceptance, the fixation of carbon dioxide continues. After regeneration of carbon dioxide acceptor, the fourth step is refixation of carbon dioxide in the bundle sheath cells. Refixation of carbon dioxide in the bundle sheath cells. Here we have generated NADPH2, but again we have carbon dioxide. So the carbon dioxide and NADPH2 that are formed in the bundle sheath have an important role to play as far as the refixation of carbon dioxide is concerned. The formed carbon dioxide is refixed in a normal C3 pathway by ribulose biphosphate. And the product here is phosphoglyceric acid or glycerate phosphate. Now, this nicotine amide adenine dinucleotide is used to reduce, it is used at the hydrogen from here, is used to reduce PGA into a triose sugar that is PGAL. So this is how carbon dioxide is fixed in the C4 plants. I mean, the initial step is fixation of carbon dioxide in the mesophyll cells, which is catalyzed by phosphoenopyruvic acid carboxylase, and the substrate for this step being phosphoenopyruvate. This produces malic acid or simply malate. And this malic acid is shunted, is transported from the mesophyll cells into the bundle sheaths, where it is used to form pyruvic acid when it undergoes oxidative decarboxylation, oxidation by dehydrogenation, and loss of carbon in the form of carbon dioxide. The third step involves the regeneration of carbon dioxide acceptor, I mean phosphoenopyruvic acid, from pyruvate. And this is achieved by phosphorylation of the pyruvate by an inorganic phosphate from ATP. The resulting product is phosphoenopyruvate. And finally, there is refixation of the produced carbon dioxide, refixation of the generated carbon dioxide by a normal C3 pathway. The substrate being ribulose biphosphate and the enzyme being ribulose biphosphate carboxylase. The product is PGA, and the NADPH2 provides hydrogen atoms that are used to reduce PGA into PGAL, a triose sugar, which is the end product of photosynthesis. My dear student, now there is a study question that describes the mechanism of carbon dioxide fixation, describe the mechanism of carbon dioxide fixation in sugar cane plant. You describe the mechanism of carbon dioxide fixation in a sugar cane plant. Remember, the examples of the C3 plants and representative members of the C4 plants. If you have those examples in your mind, it will be easy to know why a sugarcane plant and not any other plant. What is actually required, what is actually needed by the question. What the question needs you to do. Why a sugarcane plant? This should be your question of concern. That why a sugarcane plant? Why not a mango tree? Why not a banana plant? Rather, it is a sugarcane plant. Take that in your mind. Having seen that, we are going for a short break. We will be back very soon. So please stay tuned and don't go away.